Hello, welcome to module 5 of third week of our course. In this module, we will discuss about writing ODE based model for simple enzymatic reaction processes. So let us start. Enzymatic processes are ubiquitous. Most of the biochemical reactions that happens inside the cell are actually enzymatically catalyzed. So these are catalyzed reactions, so the enzyme is helping to happen the reaction at a favorable rate in normal room temperature. So if I have to write a ODE for a simple enzymatic reaction, the scheme can be like this as shown in this slide. And you have seen it earlier. S is becoming product, substrate S is becoming product P, and the enzyme E is helping in this, and the rate constant is K1 in this process. Now remember, this is the enzymatic reaction. That means we have to remember that the enzyme E is not getting used up in this reaction, because initially it will form complex with substrate, and then eventually it will be free again and the product will be formed. So the concentration of enzyme does not get changed in long run by this process. That we have to keep in mind. That means I have not to write a ODE for, if I have to write a ODE for the enzyme, I will not include this process in that ODE. Now if we keep that in mind, and if we do not look into any mechanistic aspect that how this particular enzymatic reaction is happening, then I can simply write a ODE which will be very simple as following the law of mass action. Eventually the rate of the process is equal to K1 into E, concentration of E, the enzyme concentration, and concentration of the substrate S. Now remember, using this rate I can formulate the ODEs. The rate of change of product concentration dP dt is equal to K1 into E into S. And as the product is formed and as the processes are reversible, substrate get used up. That means ds dt change in rate of change of substrate will be equal to minus K1 E into S. This minus is here because this process is reducing the amount of substrate as the product is formed. Now let us take this formulation for a reversible reaction. And reversible enzymatic reactions are very common, as we have discussed earlier in biology. See, for example, A may be a enzyme, a substrate which get phosphorylated to phospho A. So A may be a protein and its tyrosine residue may be getting phosphorylated by a enzyme E to give rise to PA, phosphorylated A. And the rate constant associated with this process I have considered as K1. As there is a kinase to phosphorylate the protein, the kinase is E, there is also phosphatase to dephosphorylated PA. So the kinase E is creating PA and the phosphatase P is removing that phosphate group and giving rise to A again. So it's a reversible process. And I want to write ODEs for this. So how I can write it? Look into this rate of forward reaction, that is A to PA, is given by K1 into E into A. K1 is the rate constant for the forward reaction, E is the concentration of the enzyme, A is the concentration of the substrate, that is the unphosphorylated molecule. Similarly, the backward reaction PA to A is catalyzed by the phosphatase. The enzyme is phosphatase here, so that is P. So rate of backward reaction is equal to K2 into P, that is the concentration of the phosphatase, into phosphorylated A, concentration of phosphorylated A. So K2 here is the rate constant for the backward reaction. So if I combine these two forward and reaction rate things, then I get the ODE for both the variable, that is for PA and A. So rate of change in concentration of phosphorylated A, dPA dt, is equal to, this is the first term is from the forward reaction because the forward reaction is forming PA. So that is K1 into E into A minus the backward reaction, because the backward reaction is removing PA and creating A. So the minus sign is there, and the term for the rest of the term is similar what I have got here for the backward reaction, that is K2 into P into PA. For A, the equations will be just the same with a minus sign, because from A, PA is formed, So dA dt is equal to minus K1 EA. I have a minus sign here, and plus, because the reverse reaction is forming A, so plus K2 into P into PA. So what I have a reversible reaction where A is getting phosphorylated to PA by an enzyme or a kinase called E, and the PA is dephosphorylated by a phosphatase P, and I have two ODs, one for PA, the other one for A. Now if I want to reduce these two number of ODs into only one, what I can do, I can consider the conservation of substrate. If you remember, many a time we use this conservation rule to reduce number of dependent variable, so we can reduce number of OD. So here I can assume that the total amount of the substrate, that is the A, remains constant, and that is AT, and it is made up of free A, which is not phosphorylated, plus phosphorylated A, and I can consider this whole sum is constant. So with time, total amount of A is not changing, some of the A is remaining as unphosphorylated A, and some of it becoming phosphorylated. So total AT is remaining constant. That means I can represent free A by AT minus PA. So I can represent free A as AT minus PA. So if I do that, then I don't need a separate OD for representing the rate of change in concentration of A. I can represent the system as a whole by the OD for PA only. So if I do that, what do I get? So in this initial equation that we have written, let me make it a bit clear. So if I take this ODE dPA dt, I have A term here. I can replace this A term by concentration of total A minus concentration of phosphorylated A. So I can replace this A. Then I get this OD, dPA dt is equal to K1 into E, the concentration of the enzyme, into AT minus PA, that is actually nothing but free A, minus K2 into P into PA. I am getting this OD from this one. And as A is now represented in terms of PA and AT is constant, I don't require this dA dt OD. So my whole system is represented by only this ODE. So what I have done, I have first written two ODEs based on the forward and backward reaction, and then I have used the rule of conservation of the substrate and reduced them into and combined them into one ODE. And this very frequently you have to do that to reduce the number of OD in your problem. Now till now what I have considered, I have not looked into mechanistic aspect of the enzymatic reaction. I have simply got inspired from the law of mass action and written down the OD. Now in some cases actually the enzymatic reaction mechanism may be critical, and that may affect the dynamics of the whole process. So in that case you cannot use a simple OD that we have discussed till now. The things may get a bit complicated. So let us look into a bit detailed mechanism. Remember, this mechanism that I will show is a generic one. There can be different variant of this also, and accordingly you may have to change your equations, mathematical formulation as a whole. So let us look at the detailed scheme as shown here in this slide. S is my substrate. S is by reacting with E and giving rise to a substrate enzyme complex. That's what happened. You have an enzyme, the substrate comes and binds to the binding pocket, the reaction pocket, to give rise to S into E complex. Now at this binding pocket, E helps S to become P. So I get a product substrate complex, that is PE. And this PE now separate out to give rise to me product and free enzyme. So this one. Now remember, all these steps we have considered at the beginning as a reversible one, because S and E is nothing but a binding process, so it is obviously reversible. Now SE gets converted to PE, that also is considered usually as reversible, and now PE dislodge and break down gives rise to free E and free P. That's also actually reversible reaction, reversible process. Now I can make certain assumptions here, which are rational assumptions, and I can reduce this system further. What I can assume? I can assume that AC and PE, this conversion, substrate enzyme complex and product enzyme complex conversion is very fast, and in fact it is very fast, and it is very difficult even in experiment to identify substrate enzymes complex and product enzyme complex separately. So usually in chemistry lab also people will consider both of them as together as a single entity and will consider them as a C complex. So I will consider this whole thing as a single thing and C as a complex. I will not separately consider them. And the next assumption, which is also very logical, is that the product does not go back and bind to enzyme again. That means I am not considering this step as reversible one. I will consider this reverse path not possible, only the forward thing. The product enzyme complex as the can breaks down into product and enzyme. So the reverse would not happen. And that actually most of the time happens because there is a faster one. So if I make these two assumptions, then I can reduce this system into a simpler structure. So what is happening now? S is binding to E in a reversible fashion to form the complex C, and the complex C is unidirectionally breaking down into product plus E. So initially I started with the detailed scheme, and then now I am from this detailed scheme I made some assumptions and I have written down a simplified scheme for the system. Now I will use this simplified system to write down the ODEs for the dynamics of this reaction system. Let us start with that. So my scheme is S is binding to E to form C. It is a reversible process. So K1 and K2 are two rate constant. C unidirectionally break into P and E, and the rate constant is K3. So I can write the first ODE representing rate of change of S, concentration of S, ds dt equal to, remember S is getting consumed in formation of C, and C can breaks down into S into E by this reverse mechanism, right? Reverse path. So the first term in my ODE for S is ds dt equal to minus K1 ES. K1 is the rate constant for the forward reaction, E is the concentration of the enzyme, S is the concentration of substrate, plus K2C, because K2C is giving the formation of S by this reversible reaction. Now let us write for the enzyme. d dt, the rate of change in concentration of E, is equal to K1 minus K1 E into S, same as the previous one in case of substrate S, because enzyme is getting consumed in formation of the complex C. So I have a minus term here, minus K1 E into S. Next, C is breaking down in both directions. In one direction here, it is giving rise to S and E, so I get back some E. On the other side, C is breaking down into P and E, that also gives rise to some free E. So I have two terms. So K2 into C is representing this reverse reaction from which C breaks and S and E is formed, and K3C is also giving rise to, K3C is representing the rate of this forward unidirectional reaction by which I also get E. So d dt is equal to minus K1 ES, that is the consumption, plus K2C plus K3C. Let us look into the ODE for the complex C. Now complex C is formed by one path and broken down by two paths. So obviously the formation is K1 into E into S, that is the forward rate process by which C is formed. I have two processes by which it breaks down. By one process it goes back to substrate enzyme, the other process goes to the product and enzyme. So I have minus K2C into, minus K3C. So dc dt has three terms. One term, the first one is for formation, and the last two terms as they have minus sign because they are consumption of C. Now let us look for the product. Remember, look into the scheme, product is formed only by this unidirectional process from C to P. So it is simple, dp dt rate of change of concentration of P is equal to K3 into C. That's all. I have written down the ODE for all these 4 terms, 4 dependent variables S, E, C and P. Now remember, in this case it is a bit different for the enzyme. In the earlier simple case, I have not considered any ODE for the enzyme because I have considered the enzyme concentration does not get changed with time. In this case, E is forming a complex, so some of the E is getting sequestered in the complex, and I have detailed mechanism, so I have considered E as a dependent variable and I have written down a ODE for E also, the enzyme also. So now proceed further and let us make some further assumption to simplify this ODE and write down the ODE representing the formation of the product. So we can make certain assumptions here. These assumptions you may have seen in biochemistry textbook. One assumption is that the substrate concentration, the amount of substrate is much more than the concentration of the enzyme. That is the first assumption. That means you are considering the enzyme is in less concentration than the substrate. And the second assumption is the key one, is that I am considering C, the complex formed between S and E, is at a quasi-steady state. That means with time the concentration of C is not changing. It is quasi-steady state because at the very beginning of the process obviously there will not be any steady state, but we assume that very fast it reaches steady state, and it is a quasi-steady state because you have multiple processes going on. C is formed, C is broken down, so I have a steady state there. The formation and breaking rate are equal, that's why I get a steady state for C. Now you can easily see that the assumption 2 requires assumption A, because if I have to continuously produce C and C is continuously broken down into P and E, then I should have constant supply of substrate, so new C molecule can be formed. So that's why you have to assume substrate concentration is much higher than enzyme concentration, otherwise I will not get the steady state of C for a tangible time. So if I have these two assumptions, then I can assume that I can consider that C is at a steady state. Now what does that mean? If C has a steady state, then this ODE dc dt becomes equal to 0. That's what I can write. Then dc dt equal to 0. That means K1 E into S minus K2 into C minus K3C is equal to 0, because that's how I have got from the ODE for dc dt. Now I will rearrange terms so that I can get C at one side and everything else on the other side. Because remember, C is the complex from which the product is formed. So I will try to separate C in one side from this algebraic relation and keep rest of the thing on the other side. So let us try that. So what I can do is I can consider what I have done, K2 and K3 both are associated with C, so I have taken them together. So I write the algebraic relation like this: K1 into E into S minus K2 plus K3 into C is equal to 0. This is very simple. This part is here. Now I have assumed a conservation. Obviously the total enzyme is conserved here because there is no production or degradation of enzyme. So I can consider that the total enzyme ET remain conserved with time, and that is equal to free enzyme plus enzyme in the complex. Right? If I consider 1:1 stoichiometry, then the concentration of the complex is equivalent to the concentration of the enzyme sequestered in that, and I consider this whole thing as equal to constant. So with time, ET is not changing, so I can replace this E in my algebraic relation using this ET minus C. That's what I will do. So I have replaced this E by ET minus C, concentration of C. So E is free enzyme and I am replacing it by ET, total enzyme, minus complex. So I get a relation K1 into ET minus C into S minus K2 plus K3 equal to C. So I again get another C here. I will take out that C and club with this C. So what I get? I take K2 plus K3 into C on the other side. Then I can rewrite this whole thing like this, taking the K1 on the other side here. So I get K2 plus K3 divided by K1. So this whole thing is a constant. These are all combination of 3 constants. Then I can write, multiply S inside this bracket. So I get Et into S minus C into S. I take C into S on the other side here on the right hand side. So I get Et into S equal to K2 plus K3 by K1 into C plus C into S. Let us separate C. So if I take C common from this, I get K2 plus K3 by K1 plus S, that goes in the denominator, and I have Et into S on the numerator. So this whole thing comes here and this one is here. So what I have got, I have separated out C and I have everything else on the right hand side. Right hand side has Et, which is constant, the total amount of enzyme, substrate which is varying with time, concentration of substrate is varying with time, divided by K2 plus K3 by K1, that as a whole is a constant because K3, K2, K and K1 are rate constant, plus concentration of the substrate. So let us clean a bit. What I have got, this is my scheme and I have initially written down the ODs. From the dc dt, I have got this relation considering steady state for C, that C is equal to Et into S divided by K2 plus K3 by K1 plus S. So now take this ODE. dp dt, there is a rate of change in concentration of the product. That's what I am bothered about here, because I want to know how with time the concentration of product is changing. So dp dt is equal to K3 into C. So I can replace this C by the C that I have considered here, because I know C is in steady state, because that's what my assumption is. So I can replace C by the steady state value of C, that is, so dp dt is equal to now K3 into Et into S divided by K2 plus K3 divided by K1 plus S. Now K2 plus K3 divided by K1 is a well-known constant, and that is called Michaelis-Menten constant. It is called Km. And remember, this is very common in biochemistry book. You will find in many a time biologists and biochemists who characterize enzyme, they calculate this value, Michaelis-Menten constant, for these enzymes, and that is very helpful for our modeling, because we can take those numerical values for Michaelis-Menten constant and plug into our ODE. So Km is defined as K2 plus K3 by K1. Now I can rewrite this ODE. dp dt is equal to K3 remain on the top numerator, K3 into Et into S, concentration of substrate. Et is the total concentration of the total enzyme, divided by, in the denominator, Km plus A. Km is the Michaelis-Menten constant, and remember this K3 is sometimes called Kcat. In some textbook you may find K3 is called Kcat. This whole ODE is essentially ODE for the product formation if I consider those two assumptions, that substrate is much more than the enzyme and C is in a quasi-steady state. And this kinetics is called Michaelis-Menten kinetics. So if I have a biological system where the substrate is in large amount, and I can safely, if I can assume that the concentration of the complex formed between substrate and enzyme is remaining in a quasi-steady state for the period of my study, I can consider that the enzymatic scheme is actually following Michaelis-Menten kinetics, and then I can use this relationship, this ODE, that dp dt, rate of product formation, is equal to K3, which some book will call Kcat, into Et into S divided by Km and A plus S. You can easily see K1 has a unit of 1 by molar and second, whereas K2 and K3 has unit 1 by second. So Km, the Michaelis-Menten's unit, is also molar. And obviously you have in the denominator substrate is in molar term and Km is also molar term, so you can sum them, because to sum two quantity their unit has to be same. Now K3 is 1 by second, 1 by time. Now, if you look into this Michaelis-Menten relationship, which is very common in any biochemistry book, you can easily see that if my enzyme is following Michaelis-Menten kinetics, then I require two parameters. One is K3, that is the Kcat, and you have to tell me Km. And if you can tell me this parameter, then depending upon how much enzyme and how much substrate I have, I can easily understand the dynamics of product formation. So usually when you are dealing with Michaelis-Menten type kinetics in a large system with multiple enzymes, you have to keep in mind that you should note these K3 and Km values for each of these enzymes. Now let us look into a scheme which is very common in biology, that is enzymatic cascade. And in any textbook of biology, you will find it very frequently. In many times, we call them phosphorylase system, the way I have shown here. So a signal comes, which may be an enzyme actually, a kinase, which activates A, which convert A to phosphorylated A, and PA is active, whereas your A is inactive enzyme. Once PA is active, PA is again a kinase, and that activates B to PB, phosphorylated to B, phosphorylated B. Phosphorylated B is again active as a kinase, and that phosphorylates C to PC. And at each of these steps, I have phosphatase P1, P2, and P3. Sometimes these phosphatase may be common. A single phosphatase may be working on both PA, PB, and PC. Whatever it is a cascade, you can see. First, one phosphorylation is happening, then that converts the enzyme into active enzyme. Inactive enzyme, which is a kinase now, will phosphorylate another substrate, which is an inactive enzyme. On phosphorylation, that will become active, and a chain of event happens, and this is a reversible reaction. Now, in these reversible processes, each of these unit are actually, I can assume, as a switch. What type of switch? This arrow representing that the input is coming from this kinase, and I can consider the concentration of this PB as output. So, the concentration of PB at a particular time is the output, and this one is my input to the switch. So, I have multiple input output switch connected together one after another in a cascade, so that this is my initial input in my system and the final output is this one. So, if I have to model this type of system, how can I approach? What I will do, I will focus now into one single unit, as this I can consider as a motif, a reversible reaction happening, and it is a switch made up of two reversible, one reversible system by which one molecule is getting phosphorylated and that is again getting dephosphorylated. So let us look into this particular motif and try to understand the dynamics of that in terms of ODE. So I have written down this motif separately. Here Y is the inactive enzyme. It gets phosphorylated by this kinase EK, and it becomes YP, which is active. I can consider it is an active enzyme, or in some case it may be inactive also, but let us consider that is as an active enzyme. EP is my phosphatase. So EK, the kinase, is converting Y to YP, phosphorylated Y, and the phosphatase EP is removing the phosphate group from YP and giving rise to Y again. And assume that both these reactions, forward and backward reaction, follow Michaelis-Menten kinetics. So they are following Michaelis-Menten kinetics. And as I said, a Michaelis-Menten kinetics has two parameters. One is the Kcat, another one is the Michaelis-Menten constant. So for this forward reaction, Kcat is suppose K1, and the second one is suppose the Michaelis-Menten is Km1. For the next reaction, the reverse reaction, K2 is the Kcat, and Km2 is my Michaelis-Menten constant. So I can write down the rate of forward reaction will be, as per Michaelis-Menten kinetics, K1 into Ek, that is the total concentration of Ek kinase, into Y, that is substrate concentration, by Michaelis-Menten constant for that reaction, plus substrate concentration. Similarly, for the reverse reaction, I have rate equal to K2 into EP into EP is the total concentration of the phosphatase into YP. YP is the concentration of the substrate here, that is the phosphorylated Y, divided by, in the denominator, Km2, that is the Michaelis-Menten constant, plus the substrate YP. Now, if I have to write down the dyP dt, the rate of change of Yp, I will club these two and I can get the equation dyP dt is equal to K1 into Ek into Y divided by Km1 plus Y minus K2 EP into Yp divided by Km2 and Yp. I have taken this one and taken this one and added minus sign here because this is for the reverse one and the other one is for the forward one. Now, Yp is the amount of free Y. I can reduce this into, I can convert this in terms of amount of total enzyme, total Y and phosphorylated Y, considering a conservation. So I can consider total amount of the substrate Yt is equal to free Y plus concentration of phosphorylated Y, and I can consider this as constant. So with time Yt is not changing. So I can replace this Y by Yt minus Yp. So that's what I have done. So if I replace Y by Yt minus Yp, I get this relation dyP dt equal to K1 Ek Yt minus Yp divided by Michaelis-Menten constant Km1 plus Yp minus Yp minus the reverse reaction rate K2 into Ep into Yp divided by Michaelis-Menten constant for the reverse reaction Km2 plus Yp. So let us clean it a bit. What I have an enzymatic switch which is a motif present in enzymatic cascade. Y is becoming Yp by a kinase Ek and it is getting dephosphorylated by Ep. Both are following Michaelis-Menten kinetics and I get a ODE like this which represent rate of change of Yp with respect to time. Now let us assume the system has reached steady state. I want to characterize the steady state behavior of this system. So that means dyP dt will be 0. So I can write dyP dt equal to 0. That means this whole algebraic relation will be equal to 0. And then I can separate out different terms on left hand side and right hand side. In this case what I will try to separate out Ek, that is the concentration of the kinase enzyme, total concentration of kinase enzyme, and Yp on both sides of my equal to sign. So I will keep Ek in one side and Yp on the other side. Remember, Ek, the concentration of the kinase enzyme can be considered here just like an input, because that Ek may have got activated by an input signal. So that input signal can be represented by the concentration of the kinase enzyme. So I want to keep that separate on the left hand side and rest of the thing including Yp on the other side. So what I can do lots of algebraic rearrangement. I have skipped them here to save time, and eventually what you get is that Ek is equal to a relationship where I have represented Yp as in terms of Yp divided by Yt. So what I have done, I have represented not just Y, I have divided Yp by Yt. So my relations become simple and it has a biological meaning also. So Yp divided by Yt. So this is nothing but fraction of Y which is phosphorylated. And in fact, a biologist can measure this Y, that is Yp by Yt, that is the fraction of the total protein Y which is phosphorylated, by different assays. For example, Western blot, by ELISA, I can actually measure not the total or real number of protein which is phosphorylated, but I can measure the fraction of the total protein which is phosphorylated. So that's why, and as for the ease of algebra also, what we have done, rather than keeping Yp separate, we have made it a ratio of Yp by Yt, and that can be done easily algebraically from this relation, and I can get this whole relation, algebraic relation that you have seen here. Now this look bit complicated, and I have done some simplification also. For example, V2 is actually nothing but K2 into Ep. Obviously K2 is a rate constant. Ep is also a constant because of the concentration of the phosphatase enzyme. J1 is another parameter. J1 include two things. One is Km1, that is the Michaelis-Menten constant for the forward reaction, the kinase reaction, and total amount of the substrate is Yt. Similarly, J2 used here, which is represented here by Km2 by Yt. So I have replaced Km2 by Yt by the parameter J2. So Km2 is the Michaelis-Menten constant for this reverse reaction, dephosphorylation, and Yt is the total amount of the substrate, Y plus Yp. This J1 and J2 will be crucial in our discussion. So we have to keep in mind these are nothing but ratio of the Michaelis-Menten constant and the total amount of the substrate, that is Yt. So what I have a algebraic relation giving Ek on left hand side, Yp by Yt, that is a fraction of phosphorylated protein on the right hand side, in terms of J1, J2 and V. So if I try to plot this Ek versus Yp by Yt, I get this type of plot. Pay attention to this plot. I have these two red lines here. I have in the horizontal axis I have Ek, and the vertical axis I have Yp by Yt, the fraction of Y phosphorylated. I have two red lines. These red lines are all coming from this algebraic relation that we have derived just now. Now this particular red line, Ek is positive, but Yp by Yt is negative. That is not possible in our system. Mathematically that may be possible, but I cannot have a negative value for fraction of the protein phosphorylated. So I will not bother about this one. Look into the other red line, this one. This is also coming from my relationship. If you plot it using a plotter, you will get that. Now here what is happening? See, here in this part, Ek is positive, that is possible. Yp by Yt is also positive, that is possible. But look into it. The values of Yp by Yt here are all greater than 1, because this line, this dotted line is representing 1. Now Yp by Yt is fraction of phosphorylated protein. So fraction of the total protein which is phosphorylated. So that cannot be ever bigger than 1. You cannot have more than the protein be phosphorylated. So the fraction can never go above 1. So that means I don't need to consider this red line. What I am left with? I am left with this blue line, which is also coming from this relationship. And if you look into this positive domain, Ek is increasing in this direction. Yp by Yt is also increasing, but it saturate, moves like a S and saturate at Yp by Yt equal to 1. And that is true, that Yp by Yt should never be greater than 1. So this is the region of my interest. So in this region, this blue line is working like a sigmoid curve, initially increasing and then saturating near 1. So what I get here from this algebraic relation is that if I remove all those not possible things, then if I plot Ek in my horizontal axis and Yp by Yt in my vertical axis, then the behavior will be a sigmoidal one, where it will saturate at value, maximum value 1. So let us look into different behavior of this one. What I have? See, if I take this motif and if I derive that relationship between Ek and Yp by Yt the way I have done, then I can make a plot like this that's shown here. Here in the horizontal axis you have the concentration of the Ek, which you can consider as an input. Yp by Yt at steady state, you can consider this as an output in your vertical axis. So then you can draw different sigmoidal type curve based on values of J1 and J2. Let us look one by one. When J1 and J2 are equal to, both of them are equal to 0.5, right? In that case, I get this pink curve. Slow, very shallow sigmoid. If I make J1 and J2 slightly smaller, from 0.5 to I make to 0.1, then I get this red curve, which is quite sigmoidal. Now make J1, J2 further small. Now I have taken J1 and J2 both equal to 0.001, very small. Then this sigmoid becomes very sharp, this blue line. So what is happening? As I am making J1 and J2 very small with respect to 1, then these sigmoidal curves are becoming very sharp and I am getting a sharp S-type thing. So when I have very small J1, J2, for example, equal to 0.001, I get this blue curve, and what will happen? Up to this near 30 from 0, if you increase Ek, there is no change of output. Output remain close to 0. Then all of a sudden, slight change in Ek will cause a sharp rise in the output, that is Yp and Yt, and I will reach the saturation. So this region shown in gray mark is the very sensitive region, we call ultra sensitive region. Here slight change in the value of Ek will sharply change the value of Yp by Yt, and then again in the rest of the region I will remain at the higher value. So this lower value I can consider as off, and the higher value I can consider as on. So when J1 and J2 are very small with respect to 1, I get a ultra sensitive on-off switch. Here you may not be seeing this one, if there is a double less than the one. So J1 and J2 is very small with respect to 1. But so in this one is like an on-off switch, the way you use in your house, on and off, two option, binary. But if you have J1 and J2 close to 1 but smaller than 1, then I get this sigmoidal thing which is shallow. That means you are increasing output slowly and input is also slowly increasing. There is no sharp jump in input-output relation. So this is like a rheostat. So depending upon the value of J1 and J2, I can have this reversible reaction working as a rheostat or as a on-off switch. Before we close, let us look into what is J1 and J2 once again. J1 and J2 is nothing but ratio of Michaelis-Menten constant and total substrate. Michaelis-Menten constant and total substrate. So when the total substrate concentration is very high with respect to Michaelis-Menten constants, then I will have J1, J2 very small with respect to 1, then I will have this on-off switch. But when the concentration of the total substrate is close to Michaelis-Menten, then I will get value of J1, J2 close to 1, then I will get this rheostat behavior. So depending on the substrate, the same input can give rise to a sharp on-off switch like the input-output relation, or it may give a rheostat type slow, shallow increase in change in dynamics in input-output. So if I jot down, if we don't need to look into the mechanistic aspect or complications in the enzymatic process, I can simply model using law of mass action for a particular enzyme reaction. But in some cases, I need to consider a complicated system, and I can assume Michaelis-Menten kinetics for those cases where substrate concentration is much higher than the enzyme and substrate enzyme complex remains as a quasi-steady state. If these two conditions are met, I can consider a Michaelis-Menten kinetics, otherwise I cannot. If I have a reversible reaction scheme which works as a switch in a large cascade, and if both the processes, the forward and reverse reaction follow Michaelis-Menten kinetics, then I can get a switch which has two behavior. When the substrate concentration is close to Michaelis-Menten constant, the switch will work like a rheostat. When the concentration of the substrates will be much higher than the Michaelis-Menten constant, the same reversible reaction switch will work as a ultra sensitive on-off switch. That's all for this module. Thank you for watching.